Got everything on display. They like to keep it interesting. So they're giving me a new challenge every day. So, we had our exam on Wednesday. How's everybody feeling about it? I heard a nice. At least one person thought it was all right. I'm busy grading those. Please be patient. Remember, there are a lot of you, and I'm doing my best to get through them. The TIs are going to help me next Tuesday, and we should be able to finish up then and hopefully get your grades posted Tuesday or Wednesday. And then you'll get them back in recitation to look at next week. You do have recitation this week. Be sure and go. All right. Any questions before we get started on lecture today? All right. We're going to continue with DNA replication. We ended on Monday with this question: what is the problem after each round of replication? So we looked at how it starts, the enzymes involved, how it proceeds. Now we want to look at how it ends. All right, so that was your hint from Monday. All right, so we're going to start out today. Quick review. Everyone pull out a piece of paper and draw a replication bubble. Get the leading and lagging strands in there. Make sure you get them going the right way. Get their artistic skills warmed up first thing in the morning. All right, has everyone had a chance to draw their replication bubble? One more second. All right, what direction is the new strand always made? 5' to 3'. Let's take a look. Your replication bubble should look something like this. It should have symmetry, mirror symmetry of sorts on either side of the replication bubble. Both sides have a leading strand. Both sides have a lagging strand, or a set of lagging strands. Both sides have a replication fork. Right, if we look at just half of that, remember that the leading strand is synthesized towards the replication fork. The lagging strand is synthesized away from the replication fork. The fragments, all right, the lagging strand is synthesize discontinuously in fragments. The leading strand is synthesized continuously as one piece. Those fragments on the lagging strand are called Okazaki fragments. We didn't talk about on Monday, but we're going to start talking about some of the differences between prokaryotes and eukaryotes and the way that replication proceeds. One of those is the size of the Okazaki fragments. In prokaryotes, they're long, up in the thousands of nucleotides. In eukaryotes, they're short, a couple of hundred nucleotides. A big difference in fragment length. So we talked about, at least in prokaryotes, all the enzymes involved. What are the major differences in enzymes involved in replication between prokaryotes and eukaryotes? types of polymerases. All right, we talked about polymerase 3 and polymerase 1 for prokaryotes. There are different types of polymerases in eukaryotes. There's also a lot more DNA. A lot more types of replication to account for. All right, so we've looked at how replication is accomplished, the enzymes involved. Today we're going to look at how replication ends. All right, so if we focus on just one side of our replication bubble, draw how each strand finishes replication. All right, think about the process and what happens at the end of the leading strand versus the lagging strand. Once you have that thought through, all right, is there a difference in the way that they end? Talk to your neighbors, figure out what the difference is between how the leading strand and the lagging strand finishes replication. And where does that start? What does it need? 3' OH. Right? It needs those. So polymerase 1 in prokaryote is going to look for those 3' hydroxyls and replace the RNA primer so that you get an end product that looks like that. Leading strand, how does it end? Just reads right off the end. Right? It doesn't have that discontinuous problem. It can synthesize right to the end. So in each one of these, we have an RNA primer on the end. What's the problem? All right, what happens to the RNA primer?
It gets removed by DNA polymerase 1.
All right, say that again,
It gets
Jessica.
removed by DNA polymerase 1 for the ligand.
Can it be removed? What does DNA polymerase 1 need to act?
3' end.
So is there a 3' in there for it to hook onto? So what happens to the RNA primer?
Replaced by DNA polymerase.
All right? If nothing else happens, what happens to our RNA primer? It gets degraded. All right, RNA isn't stable. There are enzymes in the cell designed to degrade them. It disappears. All right, nothing else happens. So why is that a problem? How do we lose information? All right, so if we continue to replicate this piece now where we've lost that little bit at the end,
The next piece will lose a little bit, so on and so forth until it.
so you lose a little bit on that end. Exactly. Replicate that piece, you lose a little bit on that end. You replicate that piece, you lose a little bit on that end. Right? I think you guys already have this figured out. This is a problem because you're shortening your chromosomes from each end. Why do we care if we lose a little bit off the end each time?
We'll run out.
We'll run out. Eventually we'll run out of DNA in our chromosomes. What is the chromosome contains? Why is it so important?
Genetic code.
It contains our genetic code. It contains genetic information. What are the discrete packets we're concerned about?
Chromosomes.
Genes. Right? There are genes on chromosomes that code for products that are turned into proteins that do things for our cell. If we continue to degrade each end, eventually we're going to start to degrade into important genes. All right. So how do chromosomes prevent themselves from getting shorter at each round of replication?
The polymerase.
Polymerase. All right. Specifically, right, if this is our piece now, and those gray bits are our RNA primer, right? So which strand is the problem?
Bottom one.
The one with RNA primer. Can the normal DNA replication machinery copy all the way out to the end of the chromosome? Okay, I heard some no's. Why not? It needs a 3' hydroxyl. But even more than that, as it gets closer to the end of the chromosome, the DNA replication machinery needs space. It just needs enough chromosome to hook onto. Right? Eventually it just runs out of nucleotides, and there's not enough room to lay down another RNA primer and to make more Okazaki fragments. All right, so you're going to lose even more of that chromosome. So we have this enzyme called telomerase, and it can come in and bind to the end of the chromosome. What's the end of the chromosome called?
Telomere.
Telomere. So telomere is the end. Telomerase is the enzyme that's going to fix this problem for us. What is unique about the end of the chromosome?
Doesn't code for anything.
Doesn't code for anything. Exactly. So it is stretches of a repeated sequence
TTAGGG.
in humans. I heard it. Alright, so that set of 6 base pairs is repeated hundreds to thousands of times at the end of each chromosome. Alright, so it acts as a barrier, protective buffer, right? It's non-coding. So as chromosomes are shortened, you're not degrading your genetic information. Also, because it is repeated, it provides a convenient, conserved binding site. So if telomerase is binding to the end of our chromosome, it's an enzyme, we know, but what must it carry with it? It's got to have a primer. So telomerase is a unique DNA polymerase. It's an enzyme, but it's carrying its own primer. that's complementary to that repeated bit of sequence at the end of the chromosome. All right, so it binds to the end of our chromosome. What happens next? What do polymerases do?
It replicates DNA.
So it replicates the DNA. Which DNA does it replicate? Which strand, purple or black?
Black. Well,
Hands for purple. Sort of halfway up there. Hands for black. Hands for not willing to commit. All right, where is the free 3' hydroxyl?
purple.
All right,
Uh,
that one on the telomerase is bent up and away. All right, so the free 3' hydroxyl is on that purple strand. It's on the template. All right, the telomerase is the polymerase and the primer. It makes its own template so that it can extend the template strand of the chromosome by adding another set of that repeated unit. All right, and it's because it's all the same repeated set of sequences, it can do it again. And again. All right? So we are making the chromosome longer. Oh, you guys already answered this one. All right, so we've extended the length of our chromosome. Now what happens? Ligase? What do we need before we can use the ligase?
polymerase 1.
We need a free 3' hydroxyl. How are we going to make it? Which polymerase? Well, first, what do we need on the template?
Primer.
We need a primer. Right, so primase. Now we've extended that chromosome long enough that the normal replication machinery can take over. Primase can come in, put down a primer. What happens next?
Then it makes more.
Which polymerase acts? Polymerase 3 can create an Okazaki fragment for us. Then what happens?
And polymerase 1.
Polymerase 1 uses that free 3' hydroxyl to replace the RNA primer. Then what happens?
Ligase.
Ligase.
But doesn't that not solve the problem at all? There's still a primer on the end.
Right, but we've extended our chromosome past the length that it was. So now we've copied it without losing any information. So if we lose that, we still don't lose information because we've made it longer than we started. All right, so all we're trying to do is preserve the length of the chromosome. So if we lose that little bit on the end now, we haven't lost any length. You're still looking confused.
Doesn't, I imagine the primer getting replaced, but I guess I don't care.
No, all we're using, so telomerase is just extending the length of the template so that the replication machinery can come in and copy what represents the end of the chromosome we started with. So we're not cutting into that information. We're still losing a little bit at the end, but because we've extended it, we haven't lost anything overall. Does everyone see that? All right, so I think that this will be a short video. Oops, that didn't sound good.
Chromosomes pose unique problems during DNA replication.
Just go over what we just talked about.
Because DNA polymerases can only elongate from a free 3' hydroxyl group, the replication machinery builds the lagging strand by a backstitching mechanism. RNA primers provide 3' hydroxyl groups at regular intervals along the lagging strand template. Whereas the leading strand elongates continuously in the 5' to 3' direction all the way to the end of the template, the lagging strand stops short of the end. Even if a final RNA primer were built at the very end of the chromosome, the lagging strand still would not be complete. The final primer would provide a 3' hydroxyl group to synthesize DNA, but the primers would later need to be removed. The 3' hydroxyl groups on adjacent DNA fragments provide starting places for replacing the RNA with DNA. However, at the end of the chromosome, there is no 3' hydroxyl group available to prime DNA synthesis. Because of this inability to replicate the ends, chromosomes would progressively shorten during each replication cycle. This end replication problem is solved by the enzyme telomerase. The ends of chromosomes contain a G-rich series of repeats, called a telomere. Telomerase recognizes the tip of an existing repeat sequence. Using an RNA template within the enzyme, telomerase elongates the parental strand in the 5' to 3' direction, and adds additional repeats as it moves down the parental strand. The lagging strand is then completed by DNA polymerase alpha, which carries a DNA primase as one of its subunits. In this way, the original information at the ends of linear chromosomes is completely copied in the new DNA.
All right, so even though we have lost that RNA primer at the end, we've extended past where we started. All right, so there are some other videos. We won't watch them. They'll be in your lecture slides if you want to go and take a look at those. So if we think about telomerase, right, this is great. It keeps us from losing information. Do all cells express and use telomerase? Do all cells use telomerase? No. Right? Really only our stem cells and our germline cells utilize telomerase, right? So that we keep that genetic information we're passing on to the next generation intact. All the rest of our cells, not a good sign. All the rest of our cells are shortened every round of replication. And at some point, when enough genetic information is degraded, it triggers cell death. Right? This is why our cells don't go on. Well, one of the reasons some people think chromosome shortening is related to cell aging, and therefore our own aging and cell death. And why stem cells can go on and divide forever and ever. All right, what happens here? Okay. All right, so telomerase is important, but it doesn't act in every cell. Yeah, you have a question, Bailey. Right, but so as long as you maintain that buffer, right, and that's why there's variation in the number of repeats, anywhere from hundreds to thousands, right? And it doesn't matter as long as you have some buffer there, how much is there, as long as you're not degrading into your genetic information.
Yeah, that's why I wouldn't this
So, and it's also limited a bit, if you think about the replication process, that S phase doesn't go on forever, so telomerase doesn't have the ability to just keep adding. There's a limited time window in which you can add those. So, good question.
What was the code on the, so like, what was on the coding for the repeats on the video was different? Was it like,
Right, so it's not the same in every species. All right, so this is in humans. I think on the video it said CTGGG. So there's some variation in this repeated sequence depending on what organism you're looking at. Yeah. Right, so the question is about, with the exception of the stem cells and the germline cells, all the rest of our body cells, our somatic cells, as it replicates, you're losing a little bit every time. You start off with some buffer, right? And that's why cells can only divide, well, one of the reasons I think cells can only divide so many times. Right? Your body cells have a lifespan. they age. At some point, they stop dividing. Some of that is thought to be related to this telomere shortening with every cell division. Sort of like a clock, right? Are there any other questions about the end of replication? Telomeres, telomerase? There's questions? Yeah, Laura. Stem cells and your germline cells. Those that are going to provide, to make gametes, and the ones that are passing on the genetic information to the next generation. You definitely don't want that information degraded. All right. So this is only a problem for things that have linear chromosomes. What don't have linear chromosomes? Bacteria. All right, we talked about bacteria at some point on Monday, I think, or Friday. We calculated just a quick back of the envelope replication rate. All right, we came up with approximately 833, around 1,000 base pairs per second for a bacterial cell with a genome of 1 million base pairs to divide every 20 minutes. All right, so it's replicating pretty fast. How long is the average human chromosome? Oh, come on. Can't win today. All right. How big is the human genome in base pairs? Anyone have any guesses? We're talking about 1 million for a bacterium. It's 3 billion. I'm impressed. It's 3 billion. That was a really good guess. It's around 3 billion.
3.4 billion?
Sorry?
Was it 3.4 billion?
3.4 billion, even better. You guys have paying attention to your human genomes. So how many chromosomes do we have in a haploid? Haploid number of chromosomes? Haploid number of chromosomes? 23. Alright, so we have 3 billion roughly base pairs. I'm going to forget the 0.4 now, it makes math difficult. And we have 23 chromosomes. So on average, how long is each of our chromosomes? I heard someone shout out a number just now. Oh, that was not it. So to basically divide 3 billion by 23 comes out to about 130 million base pairs per chromosome. Roughly, I think, if I did my math right. It's a lot of information to copy. All right, so, is that going the right way? We know the rate roughly in bacteria. How fast do eukaryotes have to copy their DNA? Not to say 3 billion. All right, that's 6 billion for our diploid number. All right. So think about that for a minute. One of the ways that eukaryotes help solve this problem, right? We talked about how long it would take. Think about if it went at the rate of, if it's moving at about the same rate as bacteria, 1,000 base pairs per second. Still think about how long it would take to get from one end of the chromosome to the other. Pretty long time. I haven't done that math, but long time. All right? So one of the ways that this is solved in eukaryotes with where replication starts. What is the location on the chromosome where replication starts called?
Bubble?
Replication fork is where the action is happening. The bubble is the form that we're trying to get to, but how does the machinery know where to start? I heard it, the origin. The origin of replication. So there's a sequence in our genomes, in bacterial genomes, that a set of proteins recognizes, called the origin of replication. And these proteins can then bind, break apart the double-stranded helix there, and then the replication machinery can come in and start acting. Right? So that replication machinery needs there to be a break in order to begin doing its thing. So our origin of replication. So we've already looked at what happens in linear chromosomes. What about in prokaryotes that have circular pieces of DNA? They also have an origin of replication. Where are the replication forks?
Where it splits.
How many are there? In what directions are they moving?
Away from each other.
Away from each other. It's moving in a circle, right? So in bacteria, you have an origin of replication. It splits apart. Just like in a linear chromosome, those replication forks open up away from each other. And new DNA is formed in each direction in leading and lagging fashion. It continues. And then gets to the other side of the circle, and there's a termination signal, right? It just needs to finish that up, and boom, it has 2 new copies. It doesn't have an end it needs to worry about. And their genomes are generally smaller, so they can get away with opening up this origin of replication and proceeding in both directions. So they run into each other on the other side of the circle. All right, what about eukaryotes? Can they do the same thing? How do eukaryotes get around the problem that they have so much genetic material to copy?
They start at more than one place.
They start at more than one place. So there are multiple origins of replication along each chromosome. So they don't have just one bubble moving away from each other. They have multiple bubbles that are going to run into each other. And these origins are every 50 to 300 kilobases. And there's, like I said, there's a protein complex, this origin recognition complex, a set of proteins designed to recognize this origin of replication, initiation sequence. How are they recognizing the initiation sequence? What are they interacting with? Is it on the last exam? The major groove, right? If a protein is looking for a specific sequence of nucleotides, it's binding in that major groove, most likely. All right, so it's scanning for this recognition sequence. It finds it in the major groove. It can open up the DNA helix at that point, creating that bubble, so the replication machinery can come in and start copying the DNA. And at each one of those replication bubbles, right, we get what we've already gone over. When in the cell cycle does replication happen? S phase. All right, are there any questions on replication? Yes. So there's basically a set of proteins called the origin recognition complex, or ORC for short. Generally, yes. Or at least many, many fewer. All right, we'll talk a bit more at the end of the semester about the cell cycle and some of the signaling in eukaryotes. It's a much more complex set of checks and balances. In prokaryotes, it's pretty simple. The recognition complex binds, opens it up, replication starts. Whoa, we still have a few minutes. In eukaryotes, There's a much more complex, something binds and then it needs to get a signal to open up so replication can start. So keep that in mind. We'll come back to it. Just briefly, I know it's Friday, everyone wants to get out of here. We're going to start next week with molecular genetics. All right, we're going to start talking about genes. So far we've gotten through DNA and replication. We're going to start talking about the other steps of the central dogma. The central dogma being a concept that describes the flow of information, the genetic information, right, in the cell. So we talked about DNA replication. We're now going to talk about how DNA gets transcribed into RNA and then translated into proteins. So our next step is transcription. I'm looking at DNA to RNA. Right, another way to say that, DNA makes RNA, RNA makes protein. And so we'll start there on Monday. You will have a short homework assignment due next Wednesday.